Hello, I am Mr. Rogers,
And I'm Mrs. Froman.
and you are watching the video notes for, we're still in section 2-3 in our book, and this now in the second half on DNA replication. So the top of your notes right now, you're going to write notes on 2-3 DNA replication. That's what I want to see on your notes when you check them. So we've talked about the cell cycle and how the process of how the cell goes from being one cell with one set of DNA. The cell is, DNA is copied and we get two cells. So now we're going to focus on that process. More is happening probably during interphase is when that DNA is copied. But how do we make copies of DNA? That's what this section notes is going to be all about. And so DNA replication gives each cell the genetic information it needs to carry out all of its activities. And this was first discovered and seen by two scientists from England named James Watson and Francis Crick. Figured out that structure. And Mrs. Froman, what is that structure of DNA here?
That's called a double helix.
Okay. You can see it here.
So double because it has two sides.
Makes sense.
And the helix, it's actually spins around, kind of like a spiral staircase.
Okay.
So DNA is a double helix. It's kind of like a twisted ladder. The sides are made of sugar, and the type of sugar is deoxyribose, which is...
Sounds familiar, like last chapter with those organic compounds that make up all of our cells. That
yes.
deoxyribose.
DNA actually stands for deoxyribose. Nucleic acid.
The acid part.
So we've got the sugar and then also phosphates, and they alternate. It goes sugar, phosphate, sugar, phosphate, sugar, phosphate, back and forth, all the way down the line.
Okay. And so these rungs then are the rungs that'd be across the middle.
Part, yep.
Are made of these nitrogen bases. So there are only 4. So think about our alphabet having 26 letters. The alphabet of DNA has only 4. And they are A for adenine, C for cytosine, T for thymine, and G for guanine. There's really only these 2 specific rules. Think about the English language. How many rules are there in the English language? A lot. Like I don't even know most of them. So, and I speak English. How about it? So, for DNA, it's really simple. There's 4 letters and 2 rules. The A for adenine always and only bonds to thymine, and the C for cytosine bonds only to guanine. So A to T and C to G.
Yeah.
And so we can see down here, we have a picture of, there's that kind of spiraled still staircase. You can see the A is always across from a T or an A. There's a C and a G. And it always follows that rule. And if we were to unwind it, and it looks more like a ladder, this is what we'd see are the, these are the phosphate bases here, right?
That's the
sugars?
Those are the sugars.
Oh, there's the phosphate. Sugar,
the
phosphate,
yellow
sugar,
things
phosphate.
are the phosphates. Yep.
And there's those all-important
and then the
bonds between them. So, from there, we're going to make some DNA. So, this is our model of what a piece of DNA might look like. So, we have our G for guanine would be there. So, opposite, G always bonds to...
It'd be C to G, so
C to G.
cytosine.
So it'd be cytosine on the opposite side. There'd be one rung on the ladder, and if I took some adenine, I'd have to put
A to T.
thymine. And if I kept going down, like if I put thymine here, that means I'd have to put adenine here.
Yeah.
And so all the letters, the A to T, C to G, they spell out the code for what makes you, and every living thing has DNA. And I think they say there's over what, like 3 billion lines of code that they've mapped out now. They kind of know all those A, T, C's, and G's. And all those lines of code, that's what makes you, is these specific combinations that tell the DNA, tell that RNA what proteins to make that make you inside your body. So these couple of basic rules that really put everything together. I'll let Mrs. Froman talk about how this actually happens, how we copy it.
So the way DNA replicates is first it has to unwind, kind of stretch itself out, and then it separates right down the middle where the 2 bases are bonded together. Yeah, like right in there. It's called a hydrogen bond, which is kind of a weak bond, so it's easy to break. So it unzips, and that little guy right there, yeah, he's unzipping the DNA. So it separates it right down the middle.
Okay.
And then you have 2 halves.
You can see, I can see like a C, there's a G on the opposite side, there's that A and the T that was on the opposite side, yeah.
And then what happens is there's little, molecules floating around in the nucleus, and they attach then to the correct thing. So if there's a T that's on the one side of the DNA ladder, then an A is going to come up and
Those
connect
guys,
to it.
and these are just kind of floating around in the nucleus, just waiting to be attached,
Yeah.
find something to attach to. Okay. You can see it down here too. So there's that floating around, the G to C,
Yeah.
T to A to T.
Yep, and they're produced by something inside the nucleus. So it's always producing those, so they're in there so that the DNA can replicate.
Okay. And I thought this was kind of cool too. We think about all the DNA in just one of your cells. So think of the cheek cells we looked at a few weeks ago. If we were to get inside the nucleus and take out all that DNA, it would stretch out 6 feet long, just not one little cell. I think that's kind of cool to think about. And so I think they think about it, if they're in the human body, there's 10 to 20 billion miles of DNA in all the cells. So we could go out to the sun and back a few times just based on the DNA in your body alone. So just the amount of information that's packed into that cell is really kind of crazy to think about.
It really is, yeah.
All right, so 7th grade, we're going to stop there. We thank you guys for watching, and we'll see you guys along the way.
Bye.